Hi everyone, welcome to Doc Hill's study where I help you get ready for your National 5 biology assessments. In this video we'll be walking through questions from an official SQA paper. The paper and the marking guide are both produced by the Scottish Qualifications Authority, the SQA, and all rights to that material belong to them. I'm using these resources strictly for educational purposes here on this non-commercial channel to support students like you in preparing for your exams. I'll be using the SQA's marking guide to highlight what answers are accepted, but keep in mind it usually only shows some of the correct answers and doesn't explain the reasoning behind them. That's where I come in. For each question, I'll walk you through the thinking process, explain what the question is really asking, and point out some of the common pitfalls to avoid. If anything doesn't make sense to you, or you want to ask about a specific point, feel free to leave a comment. I'll do my best to help you out. If you find this helpful, please give the video a like and subscribe to the channel so you don't miss any future walkthroughs. Let's get started. Okay, this is National 5 2024 Biology Section 1. This is the multiple choice paper. Let's have a look at it and see what we've got. Okay, question number 1. Let's have a read. So, cells in the stomach produce enzymes to help digest food. Identify the structure where these enzymes are produced. Okay, what structures have we got? We have structures from the ultrastructure of cells. So these are all structures within cells. So what we have to do is figure out the enzyme. So there's the key word, enzymes are produced. The rest of it doesn't matter. Enzymes are produced in all cells. So where are enzymes produced? In the cell. And the answer is going to be at the ribosome, because enzymes are proteins and the ribosome is the site of protein synthesis. Okay, good. Nice easy question to start with. Question 2. The diagram shows cells viewed under a microscope at a magnification of 100 times. Okay, we're given a number there, but I think we don't need to worry about it. Let's see. So we're given a field of view. We're given a picture of little cells. We've got a measurement across the field of view. This is a very traditional question in National 5 biology. And they're asking us what's the average length in micrometers of the cells shown. Okay, so this is quite nice. They've given us the measurement in micrometers. Sometimes you have to convert, but in this case, it's nice and easy. So all we have to do is count the number of cells. Each one of these things is representing one cell. We count the number of cells across the field of view. And we might count a couple just to make sure. And it seems that in most cases, there are pretty much 5 across the middle of the field of view. Across that, there's no point counting up here because your field of view is narrowing. You're not getting the same measurement. So we're going to divide this number by 5. Get my calculator out. Let's not make a mistake, but you could probably pick the answer. If you're good at maths, you'll pick the answer straight away because there's only one answer it can be. But let's just do it. 1200 divided by 5 equals 240. So there's your answer. It's the only answer it could have been. Okay? If you just look at those numbers, 2.4s. These are answers that you would get from different calculations using the same number. Okay, they always do that. Right, next question. The diagrams represent the movement of molecules across a cell membrane. Okay, we're looking at cell transport across the membrane here. Which cell would require the greatest number of mitochondria to allow the movement shown? Okay, well the key word here is mitochondria in that question. Underline that because we're looking at a transport mechanism that requires energy. The mitochondria are allowing the movement. So therefore, we're not talking about passive transport, we're talking about active transport. And remember that active transport, the molecules go uphill. They go from the lowest concentration to the higher concentration. They go from low to high concentration in active transport. So let's look at the diagram. Let's see if we can interpret the diagram. Well, we have a cell, we have arrows, and we have these little doofers here, which is probably molecules. Okay, so they're representing the molecules. There they go, molecules and direction of movement. So this cell seems to be in some kind of equilibrium. So there's similar numbers of molecules in and out. This cell is transporting stuff in, and it's got higher number of molecules outside compared to inside. So this is moving from high to low, which means that it's just diffusion. Okay, it's going downhill, high to low. Cell C here has got more molecules inside the cell and fewer outside, and it's going from high to low because the direction is the opposite direction of the previous one, high to low, which means that this is also diffusion. We're going downhill, high to low. So D, let's have a look at it. It must be the answer because we've pretty much rejected the first three. So let's see. We've got low concentration outside, high concentration inside. The movement is out to in. That means it's going from low concentration to high concentration. It's going uphill. This is our active transport. You need mitochondria for that. So that cell will have more mitochondria. So the answer to that is D. Okay. So far, so good. We just have to remember that what the definition of active transport is. It goes against the movement of diffusion. If things are going to diffuse anyway, you don't need to use energy to move them. All right. Question 4. A section of DNA contains a total of 6,400 bases, of which 32% are guanine. The number of adenine bases in this section of DNA is, and we've got some numbers here. Okay, this is a bit tricky. We've got to do a couple of calculations here. First of all, we need to decide what percentage of this section of DNA is adenine. Okay, so we'll know that, remember, with your complementary base pairing, G and C match up, and T and A match up, okay? And we know that 32% are guanine, which means that 32% have to be cytosine. So we're going to go 32 plus 32 equals 64, okay? That means that the rest of the bases in the molecule have to be T and A, and that makes that 36, if my sums are right. Check it in your calculator if you're not sure about it. Okay, so 36% are going to be T and A, and half of those have to be T, and half of those have to be A. So remember, this is all percentages. We're just working through the percentages first. We still have to get to a number, so we'll come to that in just a second. So this means that out of the total number of bases, 36% of them are T and A, which means that half of that, 18%, so that's going to be 18 plus 18 gives you 36, right? Check it in your calculator if you're not sure. 8 and 8, 16. That works for me. Okay, so what we need to know now is, so A equals 18%, right? So we need to figure out what 18% of 6,400 is. So that's easy. We're going to multiply. So 6,400 times 18 over 100 is going to equal our answer. And I'm just going to solve that, because if you should know that a little trick is just divide your 18 by 100, so it's 0.18 and multiply 6400. You can do that before you get your calculator out and just multiply that. So let's find out. So 6400 times 0.18 equals 1,152. It's not an easy number to come up with out of your head, but there it is, 1,152. That's the answer. And remember, you've got to put it into your answer booklet, so don't forget. You can work through this paper with a pencil, and then, because you have to transfer your answers into the into the grid with a pen. But you're allowed to use a pencil for your working out if you want to. Okay. Which diagram shows the concentration of product during a degradation reaction? Okay, which diagram shows the concentration of product during a degradation reaction? Okay, well, you have to remember that degradation is one of the types of reaction that enzymes carry out. And what do they do in a degradation reaction? Well, a substrate is converted to a product. Okay, so we have to see, it doesn't actually matter whether it's a degradation reaction or a synthesis reaction in this case, because the product is always going to be the result of the reaction. So the concentration of the product during any reaction, either synthesis or degradation, doesn't matter, is going to increase. So that one doesn't increase because it drops. This one is decreasing. This one stays the same. It has to be C. Okay, so question 5 is C. Let's see. Okay, so far so good. The only real difficulty here, apart from just knowing your definitions, was the question on the bases that required 2 steps of calculation. Let's see how we go with the next one. Question 6. The following stages occur during the process of genetic engineering. Okay, gene inserted into plasmid, plasmid cut open, plasmid extracted from the bacterial cell, gene extracted from the chromosome. This is an old chestnut because what they do is they give you a process that you should have learned and they ask you to put it into order. They'll give you several steps. Now it's very, very easy to get confused in these. Very, very easy to get confused. So what you're going to have to do is you're going to have to figure out a way of keeping your mind clear about this. So what I'm going to do over here is I'm going to decide which one of these comes first, which one comes second, which one comes third, and then I have to translate these numbers and pick one of these answers, okay? So let's see. Gene extracted from the chromosome. Well, you could do that first. Okay, we have to start with a gene. We have to get a gene out of a chromosome. Plasmid extracted from the bacterial cell. Well, we have to do that first as well because we could do these 2 things simultaneously. Now, I'm guessing that 3 and 4 are going to be separated somehow. Well, I think we've already got our answer here. Let's see. So we can shortcut there. I know these 2 things have to happen before these 2 things. And that's the only one. D is the only answer that we're getting that matches that. But we can shortcut it. Let's just go through the process though. The plasmid is cut open. That has to be the second step. These 2 are done simultaneously, and this has to be the third step. Okay, so I know that 3 and 4 have to come first. Let's look. Well, there's 4 and 3. They're coming first. So I'm going to answer D for that question. That's the simple shortcut to that question. Okay. Finally, there's a lot of writing here. Let's have a look at this. A group of scientists investigated the effect of temperature on the growth of genetically modified cells. The GM cells were grown in a nutrient solution of 20 degrees Celsius. This was repeated at 30 degrees Celsius and 40 degrees Celsius using fresh nutrient solutions at each temperature. Which of the following would improve the reliability of the results? Okay, remember for reliability, you want to make sure your experiment is repeatable and you get the same result every time you do the experiment. So normally we're going to be setting up more reactions for reliability or more experiments for reliability, right? Validity is different. We have to think about validity in a different way, but reliability is about producing reproducible results. So it's giving us extra temperatures to set up. Well, that would give us more detail in the result, but not necessarily more reliability. Set up 10 nutrient solutions containing GM cells at each temperature. Well, that sounds like we're repeating each experiment 10 times, which sounds good. Use the same concentration of nutrient at each temperature. Well, that would give us validity rather than reliability. Use the same types of GM cell at each temperature. That also gives us validity rather than reliability. So I'm going to go for B. Question 7, the answer is B. This is the only one that increases the number of experiments that we carry out, which is what reliability is. Okay? These reliability and validity questions are a little bit tricky. And some of the reasons for the answers are never entirely clear. Okay, so now we have a question on a respirometer. Remember that a respirometer is a device for measuring the respiration rate of an organism. Okay, and what you need to do is remove the carbon dioxide. So we'll see. Let's have a look at the question. A respirometer is set up to show that, as shown here, there's our apparatus, to investigate respiration in maggots. Okay, there's the maggots. Little fellas in there. Tube containing maggots. We've got a chemical to absorb the carbon dioxide they produce. Remember, respiration, we're going to use up oxygen and we're going to produce carbon dioxide. We use up the same amount of oxygen as we produce carbon dioxide, so those volumes are equivalent. But if we can absorb the carbon dioxide as the oxygen is used up, the level is going to move up the tube. Excellent. Okay, so that's nice and simple. We've got something to remove carbon dioxide, muzzle and cover. Now, sometimes in these questions, you really just want to check what you're being asked before you try and understand it too much, because there might be something really obvious. Let's see. Which of the following would be a suitable control for this investigation? All right, well, we're into controls here. And if we're going to have a control for an experiment, well, let's just look at this. The only experimental control that we need is we want a setup that's exactly the same as this. And we could have a negative control, which would include something in here that isn't maggots, but uses up the same amount of volume and maybe the same mass in that tube. So let's have a look. We're looking for something that's the same as this, but has nothing living in the tube, okay? So here we go. What have they changed here? They've moved the maggots to the wrong place. That's not going to change anything. That will give the same result as this. What have we got here? We've got glass beads instead of maggots. Is everything else the same? The muslin cover is the same. The glass beads have replaced the maggots. We've got a chemical to absorb carbon dioxide, and we have our scale and our start level. So that's a possibility. Let's see what the other options are. Muslin cover, glass beads, but we've removed the solution for absorbing carbon dioxide. So that's not a good control. And again, it's not here. So what we're going to do is we're going to choose the glass beads as a replacement of the maggots. We're removing the maggots. We're going to see what happens if you carry out exactly the same experiment with no maggots. And that provides us with what we call a negative control. So your answer is B for question 8. Question 9, excuse me. So we're going well so far. We've got 8 questions done. And remember, I'm going through this paper trying to talk out the thinking process. Some of you will go through this much faster in the actual exam, but even so, if we follow the thinking process that I follow, you'll find we'll not be too far off the amount of time it should take you to do these. Okay, chemical called colchicine. You'll not have heard of this. Again, remember in National 5 exams, what they do is they throw in stuff that you've never heard of because they're testing whether you can deal with unfamiliar information. But what they do with this unfamiliar information is they tell you about it. Okay, so the chemical called colchicine stops cells from producing spindle fibers during mitosis, right? Which prevents further stages occurring, presumably of mitosis. Could have been worded better. Which stage of mitosis would still occur when colchicine is present? Okay, so we're going to treat the cells with colchicine, and that stops them producing spindle fibers. Okay, so that stops the spindle fibers. That's what the question is telling us. And we have to think of any stage of mitosis that occurs before the spindle fibers are produced. Okay, because the spindle fibers won't happen. So let's have a look at what the options are. Chromosomes move to opposite poles of the cell. Well, you can't do that without the spindle. So that's wrong. Chromatids are pulled apart. Well, you can't do that without the spindle either, so that's wrong. Chromosomes shorten and thicken. Well, that doesn't require the spindle, and it happens beforehand, so that has to be correct. And 2 diploid cells are produced. Well, you're not going to get that because you haven't got proper cell division happening. The chromosomes aren't going to pull apart. So C is your correct answer. Chromosomes shorten and thicken. That happens before the spindle is produced. Okay, again, you're just being tested on your knowledge of the process, one step at a time, one step after the other. Okay, question 10. Which statement describes a function of the cerebrum? Okay, well, the trap or the pitfall here is you mix up the cerebrum with the cerebellum. Many students do this because the words look very similar, but they're 2 different parts of the brain. And we need to remember that the cerebrum is for memory and rational thinking. And the cerebellum is spatial awareness and balance. I've spelled spatial wrong, haven't I? Yeah, it doesn't matter. I'm going to do it again. Spatial and balance, right? So let's see. We're talking. We don't care about this thing anyway. This is what we're interested in: memory and rational reasoning, reasoning and memory. Okay. Remembering the rules of rugby. Bang. You do that with your cerebrum, not your cerebellum. Increasing heart rate during a sprint, that'll be the brainstem. Coordinating movement of muscles when playing football, that'll be your cerebellum. Coordination, spatial balance. Maintaining the balance during yoga, that's also yoga. Did I say yoga? Yoga, that's also your cerebellum. So you've been given 2 answers that require the cerebellum. And that's the trap they're trying to make you fall into. They're trying to make you fall in. They've given you 2 here. There's a wee note on how these exam questions are written, right? These both refer to the cerebellum, which is the thing you're most likely to confuse with that. So they've given you 2 of those. Now, if you weren't sure, you might say, well, they've given me 2 for 1, but then you can't have 2 answers for a multiple choice question. So if you know that the same part of the brain does that, you need to say, well, they have to be wrong. Okay? Anyway, A is the correct answer for number 10. 11. Which row in the table identifies a hormone, its site of production, and its target tissue? Okay, well, we're given hormones. Possibly. Let's have a look. Remember, let's just identify whether the first column is giving us any good information. Insulin is a hormone. That's a hormone. Glycogen. Again, this is one we're going to mix up, right? Glycogen and glucagon. Glucagon is a hormone, and glycogen is not a hormone. It's a storage molecule for sugar. And glucose itself is a sugar molecule. So we're on A or D. All right? Site of hormone production, the insulin is not produced in the liver, so that's wrong. Glucagon is produced in the pancreas, so that's correct, and its target tissue is the liver, so our answer has to be D. All right? Just a process of elimination. Eliminate, eliminate, and you've got your answer. Okay. Question 12. A study was carried out to investigate the relationship between age. I could possibly turn my camera at a bit of an angle. It seemed to be more comfortable, but anyway, we'll do that in the next video. A study was carried out to investigate the relationship between age and reaction time. The results are shown in the graph. Okay, let's have a quick look at the graph. We've got reaction time there in hundredths of a second. Yeah, increasing. Always make sure you know which way your axis is going. It's increasing. Sometimes they'll decrease, and you might make a mistake there. Check all the boxes. Okay, so we've got age 35 and under, age 36 to 55. Okay, we've got various age groups. So we've got groups, we've got males, we've got females. Okay. Which of the following conclusions is correct for these results? All right. Well, some fairly never say never, never say always in biology, right? Reaction time always increases with age for both males and females. Well, let's see if that's true. Always increases with age. Well, it's not increasing with age there for the males. That's pretty clear. Okay, that's decreasing with age, so that's wrong. Let's read the next one. Reaction time. It's always worth just going through these one at a time and making sure you can reject them. Reaction time always decreases with age for both males and females. Well, that one increases. That increases there, so that's wrong. Females have a lower reaction time than males in each group. Females have a lower reaction time. Okay, and then males have a lower reaction time than females in each group. Okay, well, there's a little bit of a trap here, I think, because you might think that lower reaction time might somehow be kind of worse, but they're really just talking numerically, right? So you have to answer this numerically. We're not talking about a poorer reaction time, because a poorer reaction time would be a longer response time. They're just talking about lower, and that's a numerical value, and they're talking about this number here. Okay, so females have a lower reaction time than males in each group. Well, they have a higher reaction time in each group, so that has to be wrong. Males have a lower reaction time than females in each age group, and that is correct. So we're going to go for D, right? There is a trap there, because you might make a judgment about reaction time, a value judgment, whereas we're really just talking about the actual number. Okay, so question 12, your answer has to be D, but there's a number of people who might well put C for that if they're not being careful and remembering that this has to refer to those numbers, right? Lower being a smaller number. Okay, lower, smaller number. Right. Question 13, the diagram shows the main parts of a flower. We're given a label to a structure. Let's see if we can remember everything. Well, there's a petal, there's an anther, there's a stamen, there's a stigma, there's a style, there's an ovary, and inside the ovary, the ovules. Okay, so there's our quick run through the structure of a flower. Which row in the table identifies structure G and the chromosomes complement of the cells it produces? Well, the ovary produces, that's an ovary, so G is the ovary, and it produces ovules, which are haploid. So it has to be A. Okay, so the answer to this question is A. The rest can be ignored. Ovary is correct, but diploid is wrong. Ovule is incorrect because it's the actual haploid cell. Okay, let's move on. Just need to know your flowers, right? And haploid, diploid differences. So the thing about biology, National 5 really is an awful lot of knowledge. You've got to learn it all. Repetition is your friend. The height of some cattle was measured. It's question 14. And the presence or absence of horns was noted. Which row in the table is correct? Okay. Presence or absence of horns was noted. So this is going to be a discrete variation because it's either present or absent, right? So that's what we're being asked here, is we're talking about the type of variation. Okay. The height of the cattle was measured, and height is always a continuous variation. Cattle are a range of different heights. They're probably fairly similar in most domestic herds because they're all genetically pretty identical, but anyway. Let's move on. So height is, we always remember that height is a continuous variation, and that something that is either there or not there is a discrete variation, and it is discrete variation is under the control of one or very few genes, and continuous variation is under the control of many genes, right? Polygenic means many genes, right? Poly equals many. Got to remember that. Okay, single gene. Okay, let's have a look. Horns. Continuous variation, no. We've decided that's discrete. So that's discrete. And this, continuous. It may be that this will just give us the answer. Those, continuous. Okay, continuous. It might be that this just gives us the answer. Let's see. Height is continuous. And that makes it polygenic, so I like the answer there. Height is discrete, so that's wrong. That's wrong. Horns were continuous is wrong, so wrong there, wrong there. Horns are discrete, that's correct, but that has to be few genes, so that's wrong. So this is all correct. We're going to go for B. So question 14, the answer is B. Question 15. In which part of a leaf are the stomata found? Okay. And you're given 3 tissues from the leaf. So the leaf is an organ. The organ of photosynthesis is made up of several tissues. You've got the lower epidermis, you've got the palisade mesophyll, you've got the spongy mesophyll, and you've got the leaf vein. And there's only one of those where the stomata is, because those cells are in the epidermis, and it's never entirely clear whether the stomata are always completely absent from the surface of the leaf, the upper epidermis, but in fact you're not given the upper epidermis as an option here. In nearly all leaves you'll find stomata in the lower epidermis, and so that's going to be your correct answer. That's wrong because that's inside the leaf, that's wrong because it's inside the leaf, and that's wrong because it's inside the leaf. Has to be A. Okay, question 16. The following factors can affect the rate of transpiration. Okay, transpiration, remember, it effectively has 2 meanings in National 5 biology. The first one is it's the overall movement of water through plants, is the transpiration flow of water through plants. And we also have this kind of specific idea of water evaporating off the leaf as transpiration as well. So they're talking here about water evaporating off the leaf. And we have 4 factors that are given that affect the rate of transpiration. Okay, they affect it. So you've got to make sure that you're noting that. Which of these factors would cause a decrease? So underline these words as you go through the paper so that you are able to keep your mind clear, because we're decreasing the rate of transpiration. Okay, so what's going to stop water evaporating? Basically, leafy shoot, not relevant. So what's going to slow down rate of transpiration? Well, that's increasing humidity is going to slow it down. Decreasing humidity is going to speed it up because the less moisture in the air, the more transpiration is going to occur. Increasing the surface area is going to speed it up, and decreasing the wind speed is going to slow it down because you're not taking away the extra humidity that comes from the plant and replacing it with dry air. So I'm going to go for 1 and 4, and that's answer D. Okay. Question 17, we're nearly there. I know this is quite a long video, but we are trying to make sure that we cover everything. Hemoglobin's ability to bind oxygen is affected by the pH of the blood. The table shows the percentage of hemoglobin bound to oxygen at different pH levels. Okay. Hemoglobin's ability to bind to oxygen is affected by pH. All right. So pH of blood, 7.1, 7.2, 7.4, 7.5. Okay, there's a trick here. We're going to have to be very, very, very careful. Very easy to get this one wrong. So, in fact, the first time I looked at this question, I got it wrong, and then a student in my class pointed out that where I was going wrong when we looked at the marking scheme. Percentage of hemoglobin bound to oxygen when the pH of blood is 7.1. So we're going to have to predict this answer here. We're trying to predict that is likely to be. Okay, so what we have to do is we have to decide what the difference is. So what have we got here? We've got 4.5. So from there to there, it's 4.5 difference, right? Either way, it's going to be 4.5 less or 4.5 more. From here to here, it is 9 difference, right? But what you have to be really careful about here is this is a jump of 2. So this is per 0.1 pH, and this is per 0.2 pH, okay? And then we want this one, which is 0.1 pH difference, right? So we're going back 1. So this is also 4.5 per 1. And so we have to change our rate here. So the decrease from here to here is just the 0.1% is 4.5. So it's going to be 4.5 is our best prediction. Okay, and I'm sure, and they give you this one. So if you take 9 off it, you'll give yourself a 51, which is not going to be the answer. What you want to do is take 4.5 off, and that's going to give you 55.5, which is the correct answer. So that's a really, really tricky question. What you have to see is that these numbers skip 1. There's no 7.3. And if you don't see that, you're not going to get that question right. Okay, so you've got to read these tables really carefully. Question 18, which row in the table identifies the substances absorbed into a structure found in the villus? All right, a structure found in the villus. Okay, we're given 2 structures, capillaries, lacteal. Okay, given 2 capillaries, 2 lacteals. Well, capillaries absorb stuff that dissolves in water, and lacteals absorb stuff that dissolves in fat, i.e. polar molecules, non-polar molecules, and the things that will dissolve in oils. All right, so let's see if we can do it. Fatty acids and glucose. Well, fatty acids are not going to dissolve in water, so they're not going to be dissolving in the capillaries, so that's wrong. Glucose is right, but fatty acids are wrong. Amino acids and glycerol. Well, amino acids are okay, but glycerol is wrong. The lacteal, okay, dissolves things that only dissolve in fat. Amino acids is wrong because they dissolve in water, and glucose is also wrong. Fatty acids and glycerol, that's correct. Our answer is D. Okay. Let's move on. All right, question 19. Diagram represents part. The diagram represents part of a woodland food web. Okay, food web. All right. Okay, we got our various levels. We've got some producers down here. We've got our consumers up here, and we've got our various levels of consumer. Predators and prey. Okay, which statement is true for the organisms in the food web? All right. Owl and stoat are in intra-specific competition. Well, owl and stoat are 2 different species, and intra means within the same species, so that's wrong. Bank vole and shrew, we don't even have to look at the table, at the diagram for that. Bank vole and shrew are in interspecific competition. Okay, this could be true. It means that these 2 things would have to be feeding off the same food, because this is only about food, okay? So let's look at bank vole and shrew. There's the bank vole there, and there's the shrew there. Well, the bank vole is eating the beech nuts, and the shrew is eating the ladybirds. So they're not in competition, so that's wrong. Ladybird is both a predator and a prey. Okay, well, here, let's look at the ladybird. It's eaten by the shrew, but it eats the greenfly, so it's a predator of greenfly and a prey of the shrew, so that's going to be correct. Let's look at the last one just as a matter of interest. Greenfly is a producer, which is wrong, of course. Okay, so that's wrong too. Good. You might not, I mean, you might see the word green and think chlorophyll, and maybe it is a producer, but it's not. Okay, so let's Move on to question 20. Question 20. The graph shows the changes in population size of a predator and its prey over 20 years. Okay. Right, there's the graph. Population, increasing axis. Okay, population size increasing in thousands. Thousands is important. We note that in the brackets. Predator is the solid line. Prey is the dashed line. Okay. Let's see. The difference in population. The difference in population sizes of the predator and its prey when each population was at its maximum is. Okay, there's a little bit of a trap here in just reading this question. That's the key word there, each population. So we want each population's peak population, each population's maximum. So let's see if we can find the maximum for the predator. There it is there. There's the maximum for the predator. Okay, and then the prey, its maximum is up here. Right? Now if you don't read that question carefully, you're going to maybe think that you have to compare them on the same time point on the graph, but you don't. It's each population was at its maximum. So we just got to be careful. And what they'll have done is they'll have given you this answer. They'll probably have given you this answer. And so if you get trapped into it, but this is the answer we're looking for. So what have we got here? Each one of these is worth 2, 62, 63, 62, 64, 66, 68, 70. Okay. So we've got 10 there, 2, 4, 6. We've got 16, and it's in thousands. So it's going to be 16,000. Question 20 is answer B. Now, very easy to get trapped in there if you don't read the question. It's this each population that's important. Okay, let's move across. Question 21. Which of the following is a biotic factor which could affect biodiversity? Well, we just have to remember that biotic means biological factor, and abiotic is non-biological factor, and a lot of students don't get this clear in their heads, and it doesn't help that they put an A in front of this as well, which actually visually makes it a wee bit even trickier. So we've got to remember we're looking at biotic factors, that word there, and soil moisture is not a biological factor, temperature is not a biological factor, carbon dioxide concentration is not a biological factor. So grazing is. I mean, it's not a very hard question, really, because even if you've got these mixed up, if you remember that one of them is biological, one of them is non-biological, 3 non-biologicals, you can't choose all 3. You can only choose the single one. So grazing has to be the answer, right? It's just an exam technique there. If you're not really sure about that, don't run away from it because you can't remember whether it's biotic or abiotic, because you can work out the answer. It's just a process. You can only pick one answer. So the non-biological one, the abiotic factors, there's 3 answers. For the biotic factor, there's only 1, so you can only pick grazing. Right, little trick for the exams there. 22, in an investigation into photosynthesis, a green plant was set up as shown and left in the light for 2 days. There we go. What have we done to the green plant? Let's have a look. We've covered it in black paper here. We have put it in a bag with a chemical to absorb carbon dioxide, and we've left one sitting out unaffected, or untreated, untested. Okay, so what's going to happen here? Well, no photosynthesis because you get no light. What's going to happen here? No photosynthesis because you get no carbon dioxide. These are the inputs for photosynthesis. The only other one that you need is water. Looks like everybody's going to get the same amount of water. So anyway, let's see. Leaves P, Q, and R were then tested for the presence of starch. In which of these leaves was starch present? Well, after a period of time, how long? 2 days. Yeah, after a period of time, these leaves here are going to have used up their starch resources because they've got no other form of energy to keep their cells alive. So they'll be using up their starch to carry out respiration. And this leaf might have produced a little bit of carbon dioxide because of respiration, but it's got the chemical that absorbs carbon dioxide. So there's no way this thing can do any photosynthesis. So it's not going to make any more glucose, and it's not going to make any more starch. This thing has no light, so it's not going to make any glucose, and therefore won't make any starch, and it will have to use up the starch to keep its cells alive in respiration. So leaf R is the one that's most likely to produce starch. So leaf R is the only one that's going to have starch, and that's going to be leaf R, so the answer is B. Okay? Question 22, the answer is B. Right. Question 23. Table shows the results of an investigation into the effect of temperature on egg-laying mites. Right, a little bit of unfamiliar information here. So let's have a look at it. Average length of egg-laying period is 36 days at 20 degrees, 27 days at 25 degrees, 18 days at 30 degrees. Okay. So the average number of eggs laid per female during the egg-laying period is 108, 108, 108. Okay. The females lay the same number of eggs, but they're going to lay more eggs per day because they've got a shorter amount of time there. As the temperature increases, the average number of eggs laid per female per day is, well, I think we've just answered that question. Let's just check it. So 108 eggs let's just do this. 108 divided by 36 equals, if you're not very good with fractions, like I'm not very good with fractions, that's 3 per day. So the females are laying 3 eggs per day, right? Cancel. Let's see, 27 divided by, 108 divided by 27 equals 4 eggs per day. Okay, by 27. And then 108 divided by 18. It's going to be a few more, isn't it? 108 divided by 18 equals 6 eggs per day. So that number is increasing, and the answer is A. Some idiot had to pick B in the past. No, these are easy mistakes to make. Okay, so you can look at this and you might think, well, I've just done the fractions, and you might just not really get your fractions in your head, and I'm not very good at it myself sometimes, just looking at a fraction like this and being able to say that this one is a smaller number than this one. So when in doubt, just calculate it out and get your actual answers and work forward from there. Okay? Good. Question 24. 2 to go. Which row in the table describes the changes that take place when fertilizer leaches into a river? Okay. So they've given us quite a lot to go on here. So algal population, light levels, bacteria numbers, oxygen concentration. All right. Well, we know that algae increase. You get algal blooms. So that's definitely right. So this is wrong and this is wrong. So we've eliminated these two. When the algae population increases, we get decrease in light levels. So that's correct. That's correct. We've got no difference yet here. Bacteria numbers increase or decrease. Okay. Well, what happens to the bacteria numbers? Well, because we've got an algae population, and that algae population is increasing to the point where it starts using up all the resources. We get algae dying and whatever, we're going to get an increase in bacteria, so that's going to be correct. That's going to be wrong, and the oxygen level will decrease, and that's going to be wrong. So the oxygen level is decreased. The correct answer is A for 24. Okay, this is a bit of a tricky one. You have to work through the process. If you've got a big bloom in algae, then what's going to happen is that the organisms underneath the algae, particularly the plants and things, are going to die. But we're also going to have algae dying more rapidly as well in the surface water. But the organisms in the plants, particularly in the lower part of the water, are going to die, and that's going to increase bacteria numbers because the bacteria are going to consume them as detritivores. And that's going to use up the oxygen in that water because bacteria will use up the oxygen during the process of decay. Okay, so the oxygen level is going to decrease. The bacterial numbers can't decrease anyway, so we don't have to worry about this one. This one gives us the answer. Last question. Oh my goodness me, it's a big one. All right, let's have a look. Plants containing toxic mercury compounds were used to paint ships. Right. Sorry, paint. There's an easy thing to not read correctly. Paints. Good job, I've got my reading glasses on, hey? Paints containing toxic mercury compounds were used to paint ships. As a result, mercury entered the marine food chains. Okay, there's a lot of information here by the looks of it. The concentration of mercury compounds in the tissues of organisms in the following marine food chain was measured. Okay, so plankton, they're eaten by aquatic insects, they're eaten by small fish, and they're eaten by big fish. Okay, which bar in the graph represents the mercury concentration of the small fish? Okay, this fellow here. So we're looking for that fellow. And what we have to do here is remember the process of bioaccumulation. So what happens is that all the plankton have a little bit of mercury in their body, but many plankton are eaten by, we've got a many to few relationship here, so many plankton are eaten by each aquatic insect. So the mercury increases in the bodies of the aquatic insects. This is what bioaccumulation is. And each aquatic insect has a certain amount of mercury in it. But the small fish will eat many aquatic insects, so a one-to-many relationship again. And each small fish will have more mercury in its body as we move along. And then the small fish have a certain amount of mercury in their body, and they're eaten by the large fish. And many small fish are eaten by one large fish. So that mercury accumulates in the large fish in an even greater amount. So, and that means it has to be the third highest, which is going to be B. Okay, really tricky little question there at the end, I think. But if you just remember bioaccumulation, we're going to low mercury in each individual plankton, high mercury in each individual fish, and go that way. Thanks for watching this walkthrough from Doc Hill Study. I hope it helped you feel more confident about tackling these kinds of questions. Remember, the answers I've used are based on the SQA marking guide, but it's always important to understand the reasoning behind them, not just memorize the words. That's why I focus on the process, not just the final answer. If you've got any questions, or if something didn't quite click, drop a comment below and I'll get back to you. If you found this helpful, give it a like, and don't forget to subscribe so you'll be notified when the next walkthrough goes up. Good luck with your revision, and I'll see you in the next one.